Deep mining of genomics data sources has become hot over the past few years. Pharma tech companies are doing this, therapeutic companies are looking for insights in non coding regions, and big pharma are doing this. But what about proteomics? Each and every single one of these companies has a proteomics arm, but they're mostly just skimming their data. But proteomics is just getting hotter as over the past 2 years, many new technologies have had IPOs. But all these efforts are missing a key tool in their workbench: deep data mining for proteomics, be it their own proprietary data or more than 100 terabytes of publicly available data in repositories. Why are they missing that? Plain and simple, because proteomics analysis is computationally challenging, and so the 100 terabytes data is in raw format, mining which is a huge challenge. And so, simple queries like, out of the millions of mutations found in genomics, which ones are observed in proteins? What are the differences between proteomes of different neurological disorders? These simple queries are almost impossible for all informatics pipelines, except mine. We have developed a high-performance computing pipeline called Bolt that we recently published in peer-reviewed journals. We have been funded by SBIR, and also our findings have been confirmed by reputed scientists globally. And now we want to take Bolt and turn the 100 terabytes raw data into the largest proteomics information database that we're calling 20,000AndMe. Just like there are 23 chromosomes, there are 20,000 expected proteins. But the true numbers of proteins and their forms can be in millions. And Bolt is the only pipeline that can look for all these millions of alterations. This can eventually become the reference for every proteomics research in the world, help build protein panels for diagnostics, as well as something that we can mine for novel therapeutics. And now I'm going to be giving a demo of our website where we have studied some large information sources in breast cancer, melanoma for new antigens, and ALS. For the breast cancer study, this is from NIH, where they studied 122 participants for breast tumors and adjacent normal breast tissue from 18 participants. Using Bolt, we find almost double the number of alterations than originally reported by this multi-million dollar NIH study, many of which follow the expected mutation load trend. This entire data can be parsed at the post-translation modification level to look for information silos like phosphorylation or glycosylation or formylation or succinylation or pyroglutamation, or it can be parsed at the sequence variant level to look for known somatic variants or population variants or novel variants. To give an example, here's a variant at the fibrinogen protein where threonine at position 331 is changing to an alanine. We can look at the Cosmic website from the Sanger Institute, which shows the same mutation and explains the importance of that, as well as we can get all the quantitative values from Bolt and also look at the raw spectra, raw data from this analysis. Looking at the high-level comparisons again, using Bolt with very little effort, not only are we able to identify 93% of all the mutations reported by this entire study, but we can find many more that may have been lost due to skimming of the data. Now moving on to new antigens for melanoma, we are finding rich classes of peptides that seem to be new antigens from 3' and 5' UTRs, from population and somatic variants, as well as from non-coding RNAs and out-of-frame translation, which have never been reported in such a comprehensive manner. These three datasets comprise less than 1% of all the publicly available data, and we can mine it for such rich information. So for us, sky's the limit.